before we get underway with stuff, I want to talk about a couple of sort of procedural aspects of what's going on in the course and the rest of the semester. Here we are on the 16th, which is the digestive system material. I didn't quite finish the respiratory stuff, so we'll actually finish that up a little bit and then go into digestive system. But we're getting very close to the end of the semester. We have 2 more weeks worth of class meetings, and then the final exam period. Oh, that's right. Today's the 16th. Next week's the 21st. That can be right. 23rd. Yeah. Okay. But yeah, so there's that week. And then I bet that's actually May 2nd and not April 30th. I must have put the Tuesday. Oh, I wonder if actually that's what it is.
May 2nd to Saturday.
Oh, okay, then that is correct. So for whatever reason, just that where it says 21 there, it's supposed to be 23. But okay, so the 30th, that's the last class meeting. It's not actually the last week of classes. The last week of classes is the one right after that. It's just Thursday of that week follows a Wednesday schedule. To deal with all of the snow days, we had to squeeze a few more Mondays in the schedule. And so back in February, a Wednesday was made into a Monday to make up for a snow day. And so that Wednesday is being replaced by making the Thursday and the last week of class is a Wednesday schedule. So we won't have class that last week of school. Okay. So anyways, we're going to get into the digestive system today. I have to finish up a little bit of respiratory stuff. And then we'll do the urinary system next week. And let me just mention while I'm thinking of it, because I'll probably forget later on today, we will be doing urinalysis next week. And if you tend to stop by the bathroom before coming to class next week, please come to class first. I'd like to give you a sample cup. Okay. I forgot to mention that with my Monday lab. And so a couple of people came in, put their stuff down, went to the bathroom, and then they came back in here and discovered that they needed to produce more. So just it helps if you keep that in mind. Okay. So that's what we'll be doing next week. And then the week after that, we're doing the reproductive system. And then the week after that, we don't have class, which I don't have represented in this, what do we call this? Schedule exactly. But between the 30th and the 14th, we don't have any class. The 14th is the final exam period for this class. It's actually the final exam period for Tuesday and or Thursday classes that meet at between 12:30 and, what would that be? 1:45 or something like that. And of course that includes us. Probably, well, actually none of you are obviously going to have anything after at 1:45. Or what would that be? It's actually 3 something is the next period, but the period does actually start at 12:30 based on the normal Tuesday, Thursday schedule of classes. So we'll be in here on the 14th, May 14th at 12:30. That's actually going to be for this class and my Tuesday class. What I probably will do, and when we get closer to this date, I'll finalize this, but I think between the 2 classes, there's more than 24 people, and there are only 24 seats in this room. So I think I'll have to stagger it a little bit. And so maybe I'll have my Tuesday class come in at 12:30, which they're used to. That's when their class starts. And then you guys will start later, probably 1:30. That's the way I usually start it. But whatever it is, that week on the 14th, that's our final exam period. During the final exam period, you're going to be taking the second lab practical. And if you go to the weekly material folders, at the end, there's a folder for practical 2 and the cumulative final, which includes the outline of the cases that you need to prepare for that practical and the online practical assignment. So all of that's just like the first practical. You're going to be taking a 50-question multiple choice test in class on the 14th, and you have an online assignment. It's 16 more questions for each of the 4 cases that it's based on. The only difference between the first practical and the second practical is just the content that it covers. The first practical is over the first half of the semester up through the cardiovascular system, and the second one is going to be on the second half of the semester. As far as lab material is concerned, and there's not really a immune system lab, so the immune system isn't covered there. It's just going to be respiratory, digestive, urinary, and reproductive systems that are covered there. And you'll notice that in the cases. If I remember correctly, there's a respiratory case, 2 digestive system cases, and then a urogenital system case, which is going to cover the urinary and the reproductive systems. So you have that stuff to do there. Now, I moved off of the schedule there, and I'm not going to go back to it because it takes a few clicks, but at the bottom there of the schedule, it says all work on Blackboard must be completed by May 14th at 12:30. Well, which is to mean before you come in to take that in-class portion of the practical. When you come in for the practical, I'll be on this computer up here, and I'll shut down access to the Blackboard site. If, in fact, I end up having you guys come in later to offset the the numbers so everybody will fit in the class, I won't shut off access for you guys until you come in for, if it's at 1:30 or whatever. So, but you need to have everything done before you come in. If you had me for A&P 1, that's different. Okay? Back last semester, and what I've done for a few semesters previously, is I made everything on Blackboard due usually the last day or the day after final exam week is over. What I've decided to do instead now is that things are due when you come in to take that in-class portion of practical. What I've found is people that put things off till the last minute don't do quite as well as people that do them on time. And so I can't stop people from putting things off to the last minute, certainly, but if the last minute comes sooner, then that's going to make people do the work a little bit more on time. So those 2 assignments there, the practical, online practical, and the cumulative final, both have due dates that are correspond to when you're going to be coming in. Okay. But everything on Blackboard needs to be done by then. You won't be able to go in and do any work afterwards. Yes.
So this is 5:14, 1:30 p.m.
So 5:14, whatever time I tell you we're going to actually meet. So I don't want to commit to 1:30 because I might decide, I don't remember the exact numbers between the 2 classes. So on May 14th, when you come in here for that practical. Okay, so the time might vary depending on what I decide in the end, but it's very easy to say just when you come in here, because when you come in here on this computer, I'm going to shut off access to Blackboard. So when you leave here, you won't be able to get back on Blackboard to do work. So it all has to be done by then. That's the way this can work. I don't think I've talked about the final here, have I? No. Okay. So those of you that have me for A&P1, it works exactly the same way. It's a 50 multiple choice question assignment. And like the other multiple choice assignments that you have, it's got a timer on it. The timer is based on 2 minutes per question, so the timer is 100 minutes, or an hour and 40 minutes, which should be more than enough time to complete this. Actually, if I already give you a 50 multiple choice question test in class on paper, I'd give you an hour to complete it, okay? Or an hour and 15 minutes per type of class we're in. So an hour and 40 is more than enough time to complete it. The questions that make up the test are coming from the pools that I use for the part 1 and part 2 assignments that you've had all semester. Okay? So you won't get any unexpected questions on that test. An easy way to prepare for that test, you can go into those assignments that you've done already and look at the questions and prepare for it that way. Now, there's a pool of questions that each one's pulled from. So like part 1 of the special senses chapter material might have a pool of 25 or 30 questions. And when you took that assignment, you got 10 of those questions. And each time you take that assignment over, you get a different set of 10 questions out of that larger pool. If you've taken those assignments multiple times, then you've probably seen most of those questions. There are too many variables to say this definitively, but probably about 5 or 6 attempts on any one of those things is going to get you to see all of the questions are in the pool. There might be a little variation depending on exactly how the random selection works from instance to instance and how many exact questions there are in a pool. Some pools are closer to 20, some pools are 30 or more questions. So there's a little variation there, but that's the idea. Now, the way that I built this test is I said the first 5 questions should be pulled out of the pools for special senses part 1 and special senses part 2. And then the next 5 questions are pulled out of the pools for autonomic part 1 and autonomic part 2. And the next 5 questions from endocrine part 1 and endocrine part 2. So you see how that works. It might not be exactly 5 questions per chapter. Whatever it is, it's got to equal to 50 questions. And there might be more than 10 sets of pools to pull from. But for whatever it is, it works out to basically like about 5 questions per chapter. And there might be 40 to 60 or more questions in the pools from the part 1 and 2 assignments for that chapter. So 5 of the questions from a larger pool will be pulled out. Any 2 instances, any 2 attempts that are made on this are probably going to look very different. If 2 people decide to go work on this at the same time, they go to a computer lab here on campus, and they pull their individual accounts up, and they both start this assignment at the same time, they probably will get completely different sets of questions, okay? Which is a little different from some of the other assignments that you've had, just because we're working with a bigger pool of questions. But probably every instance of the final that's attempted by any student is going to be completely unique. You can look at all of the possible questions, like I said, by going back to the old attempts that you've made. And if you haven't made enough attempts to see all of those questions, then you can just go in and start another attempt and look at the questions there. You might have, for instance, for the autonomic system, pulled up part 1 and got 9 out of 10 questions correct the first time through. So I'll let you go on to part 2, and you were happy with 9 out of 10. So you didn't go back and do that again. But that means you only saw 10 of the questions in that pool. So you might want to go back and look at it again. So open up another attempt, and it'll get a new set of 10 questions. Some of them might be repeats from your first attempt, and some of them might be new. And then you'll get to see more questions. It doesn't tell you which answers are correct, other than when you get it correct, obviously the one you picked is the right answer. But if you get it wrong, all you know is one of the 3 things you didn't choose is the correct answer. So if you want to get a definitive indication of which the correct answer is, you have to put an answer in and see how it gets scored. Be sure that when you're doing that, that the last attempt you make is the one that counts. That's what goes in the grade book. So if you're doing this and you get, on your last attempt, you get 6 questions that you've seen before and 4 questions that are new, and those are like the last 4 on the pool. Don't just answer those 4 and leave the other 6 blank, because then you're only going to get up to 4 points, and you don't want a 4 going in the grade book. The last attempt you make has to be the actual one. And if you start off with 9 out of 10 and you're redoing it, go ahead and make sure that you get 10 out of 10 as the final grade that goes in there. That's just going to help your grade in the end. Okay. Now, if it were that you go in and you open up an attempt and you see 6 repeat questions and 4 new questions, you don't know that that's necessarily the end of the pool. Okay. Really, the last attempt you pull up, if you want to be sure that you're getting all of them, has to be questions that you've seen all before. When you stop getting new questions, you're likely to be seeing all of the questions in the pool. And from that, you can look those over and be ready for what's going to be in the test you get. Okay. You have 3 attempts on that cumulative final. Something a little bit different about this is that it's not the last attempt that counts, but the best attempt. So if you do it once and you get 35 out of 50 correct, you're like, well, I can definitely do better than that. And you take it again, you get 42 out of 50 correct. And you're like, well, I got 8 more questions I can get a better score on. You try it again, but you only get 38 out of 50 correct on that 3rd try. The 42 is the one that counts. It's the best of the 3 scores. Also, the timer there on this is going to kick you off when it runs out. Okay. Now, an hour and 40 minutes should be more than enough time that anybody needs to complete this. If you have accommodations from the Disability Services Department saying that you need to have a time and a half on an assignment, an hour and 40 minutes is more than an hour, time and a half on an assignment based on how I give it in a normal situation. So basically everybody gets that accommodation the way that I set up these tests. If you're taking it and you don't complete it, you don't get to all 50 questions within the hour and 40 minutes, then you will be kicked off and you'll just get zeros for the remaining questions that you didn't complete. So partly that's to be sure that you prepare for the test ahead of time. The learning is really going to be in the studying. The test taking is really just a chance for you to show what you've learned. So do the preparation beforehand, and then you can get a really great score. The first time I ever did the cumulative final this way, and I explained to the students exactly what I've been telling you about here to prepare for it by looking back through the old assignments. The first person that completed it and handed it in, she had done that before coming in for the practical at the end. And so I just pulled her aside real quick, or when she handed it in, I asked her real quick, I saw that you got 50 out of 50 on your first attempt there. Would you care to tell me what you did to make sure you get a good grade like that? And she said, I did exactly what you said. I went back and looked through all the questions, and when I took the test, I knew all of the answers because I'd already looked at all the questions, and she got 50. Now, she only took 1 attempt because you don't need to take more attempts once you get 50. But if you don't get 50 points on it, go ahead and take it again. It's not going to hurt your grade. If you get a worse grade, then the better one goes in the grade book. But if you get a better grade, then it improves things. So go ahead and take advantage of the 3 attempts that are offered to you. And you should be able to get to 50 points out of 50 on that. So everybody should do great on the cumulative final, because it's just about how you prepare for it. Yeah.
Is it similar to the lab quizzes where, on the lab quizzes, on the subsequent attempt, it's pretty much the same exact questions you've already seen?
On the lab quizzes are 10 questions, and you see the exact same 10 questions over and over again. With this, it's drawing from the large pool of all of the part 1 and 2 assignments. So the second attempt you make is going to be very different from the first attempt you make. The third attempt is going to be very different from either of those 2. So it's not going to likely repeat any of the questions that you've seen. You might see 1 or 2 that are repeated, but really it's like 3 separate tests that you're going to be taking. Okay. Other questions? Okay, so again, these two are set to be due. Like when you click on it, it says its due date is May 14th. It does say, strange. It says 5:30, not 12:30. And that's because I guess when I was doing this, I was thinking about my night class, which is a 5:30 class. But anyways, this will remind you when things are done or due. I do need to switch it to say whatever time I tell you to come in. So when I get around to deciding when I'm going to actually have you come in to start taking the final, I'll make sure that says the correct time. But even if it says the incorrect time, I've told you verbally several times, all work on Blackboard has to be done before you walk in here to take the second practical. Okay, so even if it says 5:30 up there, I'm not going to say, oh yeah, I'll give you to 5:30 because it said that on the Blackboard. I'm saying it verbally here, and I think this is the 5th time I've said it. Everything has to be done before you walk into this class. Okay? And if you're not physically here right now, you have no excuse because this is being recorded. And you're listening to this and you say, oh, he said it 5 times now, 6 times now. Everything has to be done before you walk in for the final on the 14th. Okay. So when's everything due? When's it due? Come on, you can say it more forcefully than that.
5:14.
On the 14th, before
Final exam period.
you walk in for the final exam period. Okay, great. I'm doing that so emphatically because I want to be sure that you're aware of that and you don't try to pass off any excuse for not getting things done. That's a hard deadline, okay? I'm not going to give you extensions. I'm giving you a month advance notice. Well, almost a month advance notice. No, it is a month because this is 30 days in April. So 28 days from today, everything has to be done. I think that's plenty of advance notice for you to get on the ball and complete all that stuff. Okay. So, on top of that, because of that thing with the snow day and the last Thursday of the semester actually being a Wednesday, our last class meeting on, I forget what it is now, the
30th.
30th, thank you. You have 2 weeks before everything has to be completed. And so you can spend 1 o'clock to 4:20 on Thursday the 7th and make sure you get everything finished. Of course, you should be doing work outside of that time too, but you'll have plenty of time to complete all this stuff if you get started now. Don't put things off to the last minute, okay? If you sit down on the 13th to start doing this work, everything except for the practical and the cumulative final are past due. And so if you do part 1 of something that you hadn't get done in time, you've got to wait for me to check off so for you could go on to part 2. And if you do, when you do part 2, you got to wait for me to check off for you to go on to part 3. I'm not going to do that in a timely fashion, okay? You send me a note saying, I'm done with part 1, can you check off part 2? I'll get around to that sometime that day. You send a note to me saying, I'm done with part 2, can you check off part 3? I'll probably get around to that the beginning of the next morning, okay, which would be the 14th, okay? And then you say, I'm done with part 3, I'm ready for part 4. I'll do that at 12:30, okay? So don't put things off to the last minute, especially things that are dependent on other assignments, because there's that little nuisance of waiting for me to get 2 things. I'm going to be doing that for lots of different students, and I'm not going to be able to do it in a timely fashion for you. Don't put it off to the last minute. Also with that, while I'm thinking about it, if you're going to go back that you've already done to improve your grade, which I strongly suggest, any part 1, 2, or 3 assignments, well, and lab quizzes for that matter, make sure you can get the best possible grade out of that you can. Okay, you had 3 attempts on lab quizzes, and in 3 attempts of the exact same 10 questions over and over again, you really should get them all right.
Wait, the exact same 10?
Yeah, there's not a pool for those.
Okay, cool.
The questions in the quiz are just repeated over and over.
Right.
That's why you only get 3 chances and not unlimited chances. Really, you should only get 2 chances, but I'm not that hard-nosed about it. But the part 1, 2, and 3 assignments you can do over and over again. For those, if you're going back to redo things that you did before, again, it'll be after the due date. And so you have to, it won't release the score until I get in there. For those, if you've already done part 1, 2, 3, and 4 for a chapter and you're going back to do it better, do them in reverse order. Do part 3, which will cut off your access to part 4, which is okay because you've already done part 4. And then when you go back to do part 2, it'll cut off your access to part 3, but that's okay because you just got 100% on that. And then go back and do part 1, which will cut off your access to part 2, but that's okay because you just got 100% on that too. So do those in reverse order. Then you don't have to wait for me to check off part 1 before you redo part 2. Okay. If that doesn't make sense to you, I'll explain it to you one-on-one, showing things on the screen, that kind of whatever. But yes.
And for those of us that don't want to bother you, what are you planning to do grades-wise?
What do you mean you don't want to bother me?
Who has to bother you?
Why do you not want to bother me?
Well,
Because you don't think it's important enough for you to get the best grade you can in this class?
Well, I mean, I got a couple of friends. I don't like every day be like, hey, can you open me? So are you planning to do it like on the weekends, or like you're not going to do anything?
I try to get in every morning to go through and do it, but don't think you shouldn't bother me. It's my job to be bothered by you. No, that's not exactly what I mean, but it's my job to help you do as best as you can in this class. And if doing better means you tell me that I got to get on there and open up things, let me know. I might not be able to do it right away, but I know that I got to get to it. So if I did it at, say, 9 a.m. and at 9:30 you finish something up, you don't want to wait till the next day for me to go through and do it again on my own.
Well, you're typically doing it each morning and at night.
Ideally, yeah. But if you're waiting, let me know. And I'll try to get on there as soon as I can to do it. Just, I'm going to be a real, well, I'm not going to use that word since I'm recording, but a real one of those kinds of people in those last couple of days. Okay? Because I've warned everybody to do this ahead of time. And if you're finishing things up on the last couple of days and you're waiting for me to get in there, I'm going to suddenly be very busy. And in fact, I will, because it'll be final exam week for me too. But keep on top of that stuff. Okay, so I was trying to establish at the beginning where we were, and what I was hearing didn't make a whole lot of sense to me. So let me just check on some things. As I recall, I would have gotten to talking about gas exchange and showing you this picture here. And I talked about carbonic acid, carbonic anhydrase, oxygen moving in and carbon dioxide moving out, that sort of thing. This is external respiration. Did I also talk about internal respiration?
Yeah.
Yes. Okay. So this is the picture that the book has for external respiration. This is the picture that it has for internal respiration. Really, the only difference between the two, if we took the part of this picture that represents the alveolus and changed it into the representation of just some random tissue here. So we just changed this, what it looks like on that side. And we reversed the direction of all the arrows here. That would just be internal respiration. Okay. With one little annoying exception. The arrow here that's showing the reaction of oxygen binding to hemoglobin and displacing the hydrogen ion. The arrow in this picture is pointing the wrong direction. It's somewhat forgivable because all of the other arrows of that size in this picture are pointing to the left side of the picture. And so the graphic artist just probably copied this arrow, put it there, and then copied it and put it there too, and forgot to turn it around here. But really this arrow should be pointing the other way for this picture, which is external respiration. Okay. So if we wanted to change this picture for internal respiration, the alveolus would be the tissue, and oxygen, the big arrows for oxygen would be going from the blood cell to the tissue, and the big arrows for carbon dioxide would be going from the tissue to the okay, it's out in the hallway there. My watch just told me I had a phone call coming in. But anyways, so the carbon dioxide would be moving from the tissue into the blood cell, or the plasma in those two cases. Carbon dioxide would combine with water to make carbonic acid because of the carbonic anhydrase reaction, and carbonic acid would dissociate into hydrogen ions and bicarbonate ions, and the hydrogen ions would then go down here and exchange with oxygen on hemoglobin, and we'd get the protonated hemoglobin down here, and the oxygen would be available to be released. The carbon dioxide that's attached to hemoglobin would, whoops, I went up the wrong way. Carbon dioxide coming in would attach to hemoglobin, giving us the carbaminohemoglobin we see here. Okay, just the arrows should all be pointing the wrong direction, pointing the other direction, except for this one that's pointing the wrong direction as it is. Okay, so that's all that internal respiration is. What happens here, going in an exact opposite direction. In the book, the picture they have for internal respiration, they don't have anything drawn inside the red blood cell, but the big arrows are pointing the way they're supposed to be pointing. So that's new. So that's the end of that section. The next section is about transport of gases. And when I introduced this stuff originally, I talked about pulmonary ventilation, external respiration, gas transport, internal respiration, and cellular respiration, those 5 things. Okay. I had the, wait, did I show you that? Now, I showed you the presentation with the guy with the balloons and Dalton's Law. We established that before class. In that presentation, I might not have shown you this, but I thought I wrote it up on the screen. Yeah, on the screen there. But reproductive, respiratory.
You wrote the functions like ventilation,
Right, okay.
external respiration, gas transport, cellular respiration.
Yeah, so that's what I'm talking about. What I'm referencing in the presentation where the video was, which is very slowly opening up here, those 5 things that I wrote on the screen are right up here, okay? And you can't read them because it's a dark color on a black background, but it's ventilation, external respiration, transport of gases, internal respiration, and cellular respiration. The point I wanted to make with that is we just talked about external respiration and internal respiration. And in this list, the transport of gases is in between. But in the book, there's a section on gas transport that follows, nope, I said that wrong. Transport of gases that follows the gas exchange part. So internal respiration and external respiration are the same thing going in 2 different directions, chemically speaking. And the transport of gases is a little bit different. Since I have this up on the screen while, before I go back to what I was just showing you, this picture here shows us the bronchial tree. It's been plastinated. The trachea and the first and second level branches of bronchi have a translucent plastic injected into them. But then the third level of branching, which we call the segmental branches, are injected with different colors of plastic. So this branch right here gives rise to all of the smaller branches that are colored white, because all of these little bronchi that we can see filling up this region of the lung all originate from this point right here. And all of these red ones originate from this point right here, and all the blue ones from that, et cetera, et cetera. In the bronchial branching pattern, after the third level of branches, what are called segmental branches, they provide all of the bronchi for one segment of the lung. Okay, so I like this representation just so you can see how the branching pattern really is sort of segregated between the different regions. it wouldn't be possible in a picture like this, but all of the segments within one lobe, which might be these segments right here, are coming off of one of the second level or lobar bronchi. And then all of the segments in this other lobe down here are coming off of the other second level or lobar bronchus. And then, of course, everything in this lung is coming off of this first level or primary bronchus, just like everything on this side is coming here. Okay. So the branching pattern kind of segregates out into these segment regions. If something's affecting breathing in here, if you have some fluid accumulation from an illness of some sort, and this part of the lung is filled with fluid, it won't function properly, but all of the rest of the segments are going to work just fine because they are actually physically separate regions of the lungs. Okay. So since I have that up here, I just wanted to point that out. But the point I wanted to make is in this list here, which I didn't show you in the presentation, but I did write out. I might not have said pulmonary ventilation, just ventilation, but that means breathing. External and internal respiration are separated in this list by transport of gases, because when we take down oxygen in the lungs, that has to be transported to where we pass it off to the tissues. Or when we take on carbon dioxide from the tissues, that has to be transported to where we get rid of it at the lungs. So really, as far as the progression of functions, transport of gases is in between external and internal respiration. But within the chapter, they have gas exchange and transport of gases separate because the gas exchange thing, they go together pretty well. They didn't have a section that was external respiration and then transport of gases, and that section that's internal respiration. Now, the transport of gases, what I want to focus on here is how oxygen is carried in the blood. So it's done in the red blood cells through hemoglobin, and this is representation of the hemoglobin molecule, which has 4 pretty much identical subunits. They're not exactly identical. 2 of them are one type of protein, and the other 2 are another type of protein. But each one has a heme molecule in the middle that has an iron ion in the center of it. And hemoglobin is carrying oxygen because of the reaction of oxygen to that iron. And how saturated the hemoglobin gets with oxygen is represented in this curve here, called the oxygen saturation curve. On the y-axis, we have the percent oxygen saturation in the blood in hemoglobin, which is measured simply by a color sensor, color light sensor. If you've ever seen this being done, which I'm sure a lot of people have, a pulse oximeter is just a little thing that clips over the fingertip. It measures the pulse of the blood also, which is not about this. But the oximeter part of it is responsible for measuring the saturation. It shines a light into the skin, and it shines a light into the skin, and that light is reflected off of the hemoglobin, which has a color specifically based on the reaction of oxygen and iron. When hemoglobin is oxygenated, it has one color. When it's not oxygenated, it has a different color. And so you can measure the color reflection off of hemoglobin, and it'll tell you the percent concentration. Okay. The concentration or the saturation is tied to the partial pressure of oxygen in the system. Okay. Now, the partial pressure of oxygen in the lungs is around 100. Now, actually, we can kind of think of this in 2 ways. The air in the alveolus has a partial pressure of oxygen, and the oxygen in blood has its own partial pressure. The blood coming in from the right side of the heart, the deoxygenated blood, it's not completely deoxygenated, it just has a very low partial pressure. And when it gets to the alveoli, the high partial pressure of oxygen in the air will cause oxygen to move into the blood and into the red blood cell until it reaches equilibrium as best as it can. Here it shows in the lungs, the partial pressure is 100. Actually, the standard partial pressure that we can see in the air in the lungs, the alveolar oxygen, is about 104. But in the blood, as it passes through the oxygen, it usually reaches about a partial pressure of 100. And that's just because coming in, it's very low, around 40. And the difference between 104 and 40 is so great that not enough oxygen can get in to actually get it to full equilibrium. But a lot of oxygen gets in. Okay. And so it gets to about 100 millimeters of mercury partial pressure, which corresponds to 100% saturation of the hemoglobin, which is what we want. As the blood moves on from the lungs out into the body, the partial pressure is going to go down as the oxygen leaves the hemoglobin, goes into the tissues. Now, for the blood passing through a capillary bed in any given tissue in the body, just specifically that one capillary bed, coming in, the partial pressure is going to be close to 100. I think it's actually 95 at that point, but it's going to be kind of high. And the partial pressure of oxygen in the tissues is going to be around 40. So the oxygen is going to come out of the blood and into the tissue. If we look systemically, the partial pressure is not going to drop from 100 millimeters of mercury to 40 millimeters of mercury across the whole system. This is just where it's sort of the lowest point it will get, which you'll see within any given tissue. But across the whole system, the partial pressure is going to vary a little bit depending on the oxygen requirements in your system. Right now, all of you probably have an oxygen saturation somewhere around 95%. Okay, you have pretty good oxygen saturation. And that's regardless of where in the body we're looking. Well, I shouldn't say that. That's averaged across the whole body. Because you're not using a lot of oxygen. So in your tissues, you're probably not really pulling all of the oxygen out that you need. Because right now, your body doesn't need a whole lot of oxygen. You're not exerting yourself. If you were to get onto a treadmill or a stationary bike, like in a stress test at in a clinical setting, the systemic oxygen saturation would start to go down. It might get down to about 75, which would kind of correspond to a partial pressure of 40. Not precisely 75, but in that range. It shouldn't get all the way down there because, again, systemically, there's going to be some average. Somebody on a treadmill is probably going to see oxygen saturations get down into the 80 to 90% range. When somebody's oxygen saturation really goes down systemically, that might just be a sign that they don't have the cardiovascular or respiratory function that we would expect, okay? And we can see that happen. Now, I have cardiac issues, so I've done stress tests in the past, and I haven't, in fact, seen, when I get on a stress test and it's kind of close to the end of what they want to measure, I'm usually down to about 75% saturation. Based on how fast I get there or whatever, that gives them some data, and then I can get off well, I usually do the bike, but we can see that. But your average person should see saturation maybe down into the mid-80s range with exertion, because there's always going to be some oxygen in the arterial side of blood, and then you're giving up oxygen in the venous side of blood. So if you average those two together, it shouldn't get all the way down to the 40 range. Okay. That's going to happen when somebody's really exerting themselves and using up all that oxygen for their skeletal and cardiac muscle. Yeah.
You know, with exercise, perfusion improves, though. So that offsets the...
Right. Again, when somebody has good cardiovascular and respiratory health.
It's usually, like, once you dip below, once you get to the 80s in oxygen saturation, you can see extremities starting to get cyanotic, blue, just because of the lack of saturation.
Yeah, but I get to it a lot quicker because my cardiac output at rest is probably pretty low, and so my maximal cardiac output is never going to get up into the range of an average person just because of my particular cardiovascular problems. Now, this is your standard saturation curve. In the book, they have a couple of more curves below that, which show a couple of things that will alter saturation. And I just want to show you this one right here, which is the effective pH, okay, pH of the blood. Blood pH should be about 7.4. 7.35 is kind of the average I usually work with, but about 7.4. If pH goes down just a little bit, the saturation curve gets a little bit lower. And if pH goes up, the saturation curve goes up. Okay. So pH is going to affect the relationship of partial pressure of oxygen to saturation. Now, one of the reasons why that's true is because pH is almost precisely tied to partial pressure of carbon dioxide. We go back to the, this one right here, I do have it open. We go back here. This is external respiration, but let's think about internal respiration. As carbon dioxide's coming in, that's going to make more carbonic anhydrase, which is going to make more hydrogen ions, which are going to combine with the hemoglobin more. Or carbon dioxide will combine with hemoglobin more also. So there's less available hemoglobin to be saturated with oxygen. And the pH is tied directly to the concentration of hydrogen ions. So this reaction here, when carbon dioxide levels are high, we're going to get high hydrogen ion levels and therefore low pH. And the interactions with hemoglobin will show us a mechanism where it can interact with that a little bit. So this drop in saturation with pH can be tied to that. The respiratory system is concentrating more on keeping pH normal, and that basically means keeping carbon dioxide levels normal. The respiratory system doesn't really deal with oxygen levels directly. When you're exerting yourself and you're using up oxygen, you're also accumulating more carbon dioxide. And so the regions of your brain that regulate respiratory function are really trying to clear out carbon dioxide more than get oxygen in. It just happens to be that when you exhale carbon dioxide, you're going to follow that by inhaling oxygen. The actual systems that look at oxygen homeostasis are really controlling the production of red blood cells, which can happen. If your oxygen levels are low, then your bone marrow will be stimulated to make more red blood cells. But that takes a little bit of time. The direct changes in respiratory function is really dealing with the pH of the blood. And as the pH of the blood rises, that's going to help get oxygen saturation up to some degree. Now, did I talk about the parts of the brainstem that control breathing last time? Okay. At some point with some of these classes, I ended up talking about it earlier on, but really I tend to save it for this point. I think it's in the process of breathing section. This picture here is showing you the connections from the brainstem to the muscles that control respiration. In the medulla, there are two structures, one in a posterior position called the dorsal respiratory group, and the other in an anterior position called the ventral respiratory group, which directly cause contraction of the muscles that have to do with respiration. The dorsal respiratory group controls the diaphragm and the external intercostals. And then the ventral respiratory group controls everything else. For basic breathing, what you're doing right now, only the diaphragm and external intercostals are required. So that means that only the dorsal respiratory group is really doing any work driving breathing. Okay. It sends a signal down causing contraction of those muscles. When they contract, they expand the rib cage, the thoracic cavity volume, which enlarges the volume of the lungs, and you pull air in. When they relax, then the elastic tissue in the lungs recoils, pulling the lungs back down to their original size, and that pushes air out of the lungs. Okay. That's basic breathing. It's just this simple repeating stimulation of the DRG of those muscle groups, the diaphragm and external intercostals. That part of the brain is actually autorhythmic. It generates its action potentials without any outside input. Where have we heard that happening before?
Cardiac.
Right, in the heart. The SA node and the AV node and those sort of things are autorhythmic. They generate an action potential, and they never hit a resting potential afterwards. Instead, they slowly depolarize back up to threshold. Okay? And the rate at which that depolarization takes place sets the rhythm that the SA node's firing, the heart rate's going. Here, the same thing's happening. These cells are slowly depolarizing back to threshold. They just do it a lot slower. Typically, we talk about the cycle taking about 5 seconds per breath. So the dorsal respiratory group sends an action potential down, or action potentials down its nerve to cause contraction. And then over 5 seconds, that causes inhalation, and when it relaxes, exhalation. And then another signal comes along the next 5 seconds, causing it all happen again. If this is cycling through every 5 seconds, what's the respiratory rate that we would expect to see from that?
One complete breath every 10 seconds.
One complete breath every 5 seconds. Okay. You make those muscles contract, and when they relax, you exhale. And then 5 seconds later, there's a signal causing them to contract and then relax. So what's the respiratory rate that's set up there? How many times does that happen in a minute? How many 5 second?
12.
Yeah.
12 per minute.
So 12 times 5 equals 60. So there's 12 breaths a minute. That's not necessarily the resting respiratory rate. I guess that's what you call it. But that's usually what we think of as the regular one. There's a bit of a range depending on exactly what you're doing. But that's set up by the autorhythmic cells in the dorsal respiratory group. When you need to take a deeper breath, a signal can be sent from the dorsal respiratory group to the ventral respiratory group, causing other muscles to play their part to take a deeper breath or to exhale more quickly, which will also set up some changes for respiratory rate there too. Those kind of changes are usually established by a couple of nuclei in the pons called the pontine respiratory group. There's a pneumotaxic center and apneustic center. One inhibits and the other excites the dorsal respiratory group, therefore either increasing or decreasing respiratory rate. And then what's not pictured here but explained in the text is that the pons can get input from the hypothalamus, which is right here in this picture, or from the cerebral cortex, which would be up here in the upper part of the picture, to help regulate changes in breathing based on either higher cognitive processes or broader homeostatic mechanisms. If you need to speak, then you need to force air out across your vocal cords in a different fashion than regular breathing. So the cortex, which is where you're going to develop the impetus to speak, is going to send signals down through the pons to cause the medullary structures to control breathing so that we can breathe out and produce speech sounds. Other aspects of homeostasis might be relegated through the hypothalamus, which will change things there too. For instance, when you go to sleep, your respiratory rate changes. And so your hypothalamus is getting information about the fact that it's nighttime and you're going to sleep, whether it's tied to circadian rhythms or to other parts of the brain that control wakefulness and that kind of thing or whatever. Yeah.
Between the pneumotaxic and apneustic pontine centers there, which excited and which inhibited?
So in the picture here, you can see the line going from the pneumotaxic to the dorsal-respiratory group has a negative sign on there, meaning that it inhibits. And the apneustic has a positive sign next to its line, arrow there. So that's just there. I don't have too much to say about those two areas. I've never really looked at them that in depth. But in answer to your question, just right there in the figure, they lay it out there. Also, influencing these, which is not in the picture but is explained in the text, is there's input to the medulla directly from the periphery. Wrong direction. Before this figure, this table is pointing out different players in what's going on here. And the aortic body and the carotid body, these two structures here, those are sensory neurons in the aorta or the carotid artery, which sense the partial pressure of carbon dioxide. It says partial pressure of oxygen here, although that's not their primary focus. And then pH. pH and partial pressure of carbon dioxide are really tied closely together. And so these things will actually send signals into the dorsal respiratory group or ventral respiratory group to control the muscles of respiration. If your blood pH gets too low, it's going to cause you to exhale air more forcefully and more quickly to clear carbon dioxide out, which is going to pull more oxygen in, although that's sort of secondary to what it's actually regulating. Yeah.
So according to that picture, the DRG innervates the VRG, then because there's no line drawn from the pontine respiratory center to the VRG.
Right. So the DRG is the autorhythmic part that controls basic breathing. It gets input from higher centers, the pons and on up. When it's getting signals to do something different, it can then send signals over to the ventral respiratory group to get it activated and tied in with this. But the diaphragm and external intercostals are the primary muscles of breathing, and so that's what it controls. And when more muscles are needed, it will recruit the other respiratory group to do its job there. Okay. So yeah, there's this little arrow between the two in the figure showing the dorsal respiratory group telling the ventral respiratory group what to do. Okay, so, which might be kind of tough to see in that picture, but yeah, that's true. This goes on to talk about things that can influence respiratory rate. In bold-faced up here, it has central chemoreceptors and peripheral chemoreceptors. So I pointed out the aortic and carotid bodies. That's where some of the peripheral chemoreceptors are located. But then central chemoreceptors are talking about chemoreceptors in the brain that can pay attention to the chemical composition of blood as it pumps through the brain and send signals down itself. Okay. So that's the end of this section on breathing, which is where the book actually deals with the regulation of things. But I leave that for the end because it has a lot to do with regulating the level of gases in the blood, which we just talked about with the transport of gases stuff. There is a section here, I'm not going to say a whole lot about it, but it says modifications to respiratory functions, and it mentions a few things. This is kind of interesting in here. There's a table talking about the changes in partial pressure of oxygen at different altitudes. So New York City would be at sea level, which is effectively where we are, although actually we're a couple hundred feet above sea level here. So atmospheric pressure, 760 millimeters of mercury, and the partial pressure of oxygen in the air around us would be 159 millimeters of mercury. By the time we inhale, the partial pressure of oxygen decreases because it changes once it gets into the lungs. In Boulder, which is about 5,000 feet above sea level, therefore the mile-high city, the atmospheric pressure is dropped to 632. Partial pressure of oxygen in the air around somebody is 133. Once it gets into the lungs, it's probably a lot less than that. It's 104 in the lungs at sea level, so it's probably less than 100 in the lungs in Boulder. And then at Aspen, where we're a few thousand feet higher, the total pressure is down and the partial pressure of oxygen is down, and it's therefore going to be lower in the lungs too. And then Pikes Peak, just to stick with the Colorado thing, is probably the highest point in Colorado. Its overall pressure and its partial pressure, which is now less than 100 out in the air before we breathe it in. And then Denali, which I guess is the highest point in the Americas at 20,000, its partial pressure is here. And then Mount Everest and its overall elevation and the levels there. As you go up in altitude, the amount of oxygen present in the air, the pressure, partial pressure of it's going to go down just because the overall pressure goes down. Air pressure decreases as we get higher and higher. And there's a point at which the partial pressure of oxygen in the air is going to be too low to make proper use of and get oxygen into your system. People who live in Boulder, or I suppose Aspen, probably have sort of some homeostatic mechanisms that have worked with this so that they can do a better job of getting oxygen into their system. They might have a higher hematocrit number or something like that, I don't know. And then when people are climbing these mountains, there's a point at which oxygen levels are really low and they're probably going to need to take supplemental oxygen with them so that they can get the right amount of oxygen in, because of course they're using their skeletal muscles quite a bit to climb the mountains and they need oxygen to do that. There's a point at which, and I can't remember offhand what it is, but there's an elevation that's referred to as, I think, the dead zone, because the level of oxygen in the air is too low for life to exist. And it is somewhere along Mount Everest. So there's a point when you're climbing Mount Everest that if you go any higher, you need to have supplemental oxygen. There won't be enough for you to just survive on your own. And I don't know if there's a point on Denali or Pikes Peak like that too. I don't know the exact elevation where the partial pressure of oxygen in the air around us is too low for us to get anything into our body effectively, but it's got to be somewhere there. Okay. So just some things to think about for respiratory function. I'm not going to talk about the embryonic development stuff at all. I might have mentioned that at some point, just fetal respiration is different from adult respiration or newborn respiration even, because the fact that a fetus isn't breathing air in, it's getting its oxygen through the placental transfer. So that wraps up the respiratory system. Now I want to move on to the digestive system. Now, I am running behind on things, obviously, because I didn't finish up last week's material on time. So I'm going to try to cover as much stuff about the digestive system today. And I'm necessarily going to cover a few things a little bit overly briefly, maybe. So please do pay attention to the source material here for some of the more in-depth understanding. But In the digestive system, it starts off with talking about the anatomy of the system and sort of an overview of its functions. And then it starts talking about the different regions. So the mouth and throat, the stomach, the intestines, some accessory organs, and then some broader implications of important functions within the system. For the anatomy of the system, there's two things to address here. One is the particular organs that we have, and then I want to talk a little bit about the histology of one of those types of organs. So this picture shows all of the organs of the system, but I want to switch over to a screen where I can write this down. And I'll come back to the picture to point a few things out in a little while. But in the digestive system, there are two sets of organs. The first set is referred to as the digestive tract, which is also known as the alimentary canal, which is the term that the book uses, or the gastrointestinal tract. Gastrointestinal is abbreviated GI. So I want to talk about the structures that we see there. So I'll leave some spaces to add in there. And then the second set of organs are called the accessory organs, or accessory digestive organs. And these are what secrete digestive juices, which I'll explain when we get to those. And there are a few of those that I want to talk about too. So first, let me get at the digestive tract. This is essentially the organs that food actually moves through. Okay? And that starts with the oral cavity, which is not an organ itself. It's obviously a cavity, but it contains organs that help with digestion. There's the teeth, the tongue, and then what makes up the wall of the cavity, the muscular facial structures like the lips and the cheeks, which help us to get food into our mouth and to get it in between our teeth to chew. So all of that's in there. So the oral cavity contains the tongue, teeth, lips, and cheeks. Contain is not the best word for that, but those are the structures associated with the oral cavity, which we can think of. And they would therefore be more like organs. A muscle is an organ, or a tooth is an organ in that sense. Okay. The food then moves down into the pharynx, which we've met before because of the respiratory tract. But specifically, it's just the oro and laryngopharynx. Okay. The nasopharynx is not part of the digestive tract. You don't want food going up into your nose. It shouldn't. The soft palate should move back against the pharynx to keep food from going up in your nasal cavity. But there are situations where that doesn't work. If you're trying to do something respiratory and something digestive at the same time, like you're trying to drink your milk and laugh at a joke that the guy at the cafeteria table next to you said, then the soft palate gets confused and the milk goes out your nose. Okay, it's not a perfect system, but it's not supposed to be part of the digestive tract. And then from the pharynx, the food goes down into the esophagus, which is really just a tube to connect to the stomach. And then we go into the small intestine, which is made up of a few different parts. There's the duodenum, jejunum, and ileum. I think duodenum technically should be pronounced duodenum from a Latin perspective, but everybody pronounces it duodenum. And then there's the large intestine, which starts with the cecum. And then we have the colon, which has an ascending, transverse, descending, and sigmoid section. Blah, 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 blah. Oftentimes, colon and large intestine are used interchangeably, but those regions there are the ones that are actually called colon. And after the colon, we get to the rectum and the anus, which finishes out the large intestine. And that would, of course, be the end of the digestive tract. Now, like the respiratory tract, there's usually a separation of this into an upper and lower part. And the dividing line for that I want to put right here. Okay. So everything above this line, the esophagus, pharynx, and oral cavity, is the upper digestive system or upper GI, and everything below it is the lower GI. And that's really an anatomical distinction. Okay. Where I put that double line there, what does that actually represent? What physical structure is right there between the stomach and the esophagus? More importantly than that, what's there? Big thing separating two parts of the body from each other. The diaphragm, right? Okay. Yes, there is in fact a sphincter between the esophagus and the stomach, but the point being here is that the diaphragm as a border between the thoracic and abdominal cavities is really what's separating these two things out. There's another aspect to this, another anatomical feature that separates the two into upper and lower, which has to do with the tissues that we find lining the tract, which I'll show you in a little bit. But they transition at the same point between the esophagus and the stomach. Now, this is an anatomical definition for upper versus lower. Okay. Upper GI sometimes has a different connotation. If you hear upper GI, what's another meaning for that? Somebody says that they're scheduled for an upper GI. What does that mean? Endoscopy. And they're getting an endoscopy done. A camera is inserted through the oral cavity. It goes down the pharynx, down the esophagus. It will probably continue on down into the stomach. Okay. So clinically speaking, an upper GI usually includes looking at the stomach and maybe even just a little bit of the small intestine. A lower GI endoscopy would be a camera going in the other end. And that's more commonly referred to as what? Yeah, colonoscopy, which is only looking at the large intestine. There's a valve between the small and large intestine, which you don't really want to force something back through, like a camera, because that'll push contents of the large intestine into the small intestine. You don't want that to happen. So to be able to visualize inside the stomach, you perform an upper GI endoscopy. So from a clinical perspective, the stomach is included in that upper GI visualization. And a lower GI is really just looking at the colon. So there's different ways of defining upper and lower GI. And I've just described the anatomical and the clinical ways of saying that. If you look up online the definition of upper versus lower GI, sources will tell you that the division is actually somewhere in the small intestine because of the embryology of the system. Embryologically, the end of the small intestine and the large intestine develop out of a different part of the embryo than the rest of the tract. So it's usually laid out there. Two reasons why I want to ignore that. One, which are kind of the same reason, but one is that it's about embryology, and we don't study embryology here, okay? And two is it's not about anatomy and physiology, and we do study anatomy and physiology here, okay? So the anatomical definition I'm making here is a better description of upper and lower, but I do want to let you understand that sometimes in a clinical setting, upper GI is going to include the stomach there. So down to the accessory organs. These are organs that secrete digestive juices. Now, there are different ways of defining accessory organs, but this is the most common way. And so that's going to mean that we're talking about the salivary glands, of which there are three: the parotid, sublingual, and submandibular. And then there's the liver, which secretes a digestive juice called bile. And the gallbladder, which actually doesn't secrete digestive juice, it stores the bile to be released. It stores bile between meals to be released when it's needed to digest food. And there's the pancreas, which secretes a bunch of digestive juices too. There are different ways of interpreting what an accessory organ might be. So you might see sources that include other things, but the book and a lot of sources will stick to this list because these are the things that are secreting the digestive juices, like the saliva and bile and the pancreatic juices, into the digestive tract. Where they do that depends on, what part of the tract they're connected to, but those are the major accessory organs. So, I did not do what I wanted to do. Oh, that's right. Okay, so that's listing the digestive tract organs and the accessory organs overall. Now that I've listed them, let me go back over to this picture. That's not it. That's not it. That's not it. Oh, I never actually opened it up. I'm not going to bother opening it up. But so the oral cavity is up here, obviously where your mouth is. We see the parotid, submandibular, and sublingual salivary glands arrayed around that. And then the pharynx and esophagus carrying food down to the stomach, which we have here. And then at the end of the stomach, the small intestine begins as the duodenum, and then the jejunum and the ileum all down here, which eventually ends where it connects to the large intestine here. At the level of the duodenum, the pancreas, which is actually posterior to the stomach, right here, connects to the digestive tract. And then the liver and gallbladder connect to the digestive tract there also. So the bile and pancreatic juices that are secreted from these structures go into the duodenum to help with digestion in the small intestine. And then the large intestine starts here. So this is the cecum. It ascends, goes across as a transverse colon, descends, and then it curves around as a sigmoid colon here, with the rectum and anus finishing up the tract. So that's just a quick path through the digestive organs there. The other anatomical point to make is about the structure of digestive organ walls, the histology of these things. Now this picture, which is kind of the standard way of presenting this, is really a small intestine picture. And it's a great example of the basic structure that we're talking about here. But this applies to the other organs also. Just the other organs look a little bit different. There are 4 different layers of tissue, which are in bold-faced here. Starting at the lumen, or where the tissue is up against the food that's passing through, we have the mucosa, which is made up of an epithelium, connective tissue called the lamina propria, and a thin muscular layer, which is only a couple of fibers thick, called the muscularis mucosae. And then the next layer is called the submucosa, which is connective tissue that's just adjacent to the mucosa. There are glands in the submucosa which connect to the surface here. So this thing here shows a gland that's actually extended into the submucosa area, but the duct connects to the mucosal surface, as opposed to these glands that are completely contained within the mucosa, or a gland represented by this duct here, which is outside of the tract. So this might be a duct coming from the pancreas or the liver, gallbladder area or something like that. So that's the mucosa and the submucosa. And then beyond the mucosa, the next layer is called the muscularis, which is muscular fibers. They're arranged in layers. The first layer is the circular muscle, which wraps around the tube, and when it contracts, it constricts the tube. It pinches in on the tube. And then the other layer are longitudinal fibers, which run the length of the muscle, the length of the organ. So when they contract, they will shorten the tube up a little bit. And they mostly are going to be part of helping to move food along the length of the tube. And we'll see a representation of that later on. And then the last part is the serosa, which is a simple squamous epithelium and its associated connective tissue surrounding the outside of the organ. And like the serous membranes that we saw with the heart and the lungs, the serosa also extends away from the organ to line the inside space of the cavity where we find it. Now, the serosa is only found around the stomach and the intestines, which means it's only lining the abdominal cavity. The esophagus and structures superior to that don't have a serosa. But an important thing that the serosa does is it extends away from the organ and goes to the body wall. And in that extension, which is called the mesentery, it contains blood vessels and nerves and lymphatic vessels that are going to or coming from the organ itself. And that's represented here. A couple of other differences between what we see here, which is a small intestine again, and some of the other organs. Besides the serosa, which is only found in the lower respiratory tract, the muscularis is different between the stomach and the intestines and the esophagus. The stomach has a third layer of tissue. Instead of just circular and longitudinal, it also has a layer called oblique, which are fibers at an angle that can contract a little bit differently. Okay, and when we get to talking about the stomach, I'll explain what that's about in a bit. Also, another difference to point out is the epithelium that lines the lumen of the tract, the mucosal epithelium, comes in 2 different types. And this is the other thing that separates upper from lower digestive tract. In a lower digestive tract, meaning the stomach and the intestines, that epithelium is a simple columnar epithelium. In the upper digestive tract, which is the esophagus and the pharynx and the oral cavity, that epithelium is a stratified squamous epithelium, okay, which I introduced with the respiratory tract last time. So going back here, besides this line representing the diaphragm, also we have stratified squamous in where's that character? Whoops. In the upper respiratory tract. I put a little caret up there, like an arrow pointing up. And we have simple columnar in the lower digestive tract. And I used a lowercase v, which is like an upside down arrow. Okay. So keep in mind that this character here represents an arrow pointing up, meaning above the diaphragm. And this represents an arrow pointing down, meaning below the diaphragm. Those are the epithelial that we see there. Yes.
Yeah, diaphragm's anterior to the esophagus and stomach at that point.
No. There's actually a hole in the diaphragm called the hiatus that the esophagus passes through. So it's attached posteriorly and anteriorly to the body wall, and the esophagus passes through it. And actually, the fact that the esophagus passes through it means that the diaphragm can help limit things moving from the stomach back into the esophagus. Every time you breathe, it's going to pinch the esophagus off. So stomach contents are not going to easily move in. So the sphincter that you mentioned earlier helps keep things from going back into the esophagus, but then also the diaphragm at the hiatus, with each breath, pinches off a little bit and helps to keep things from moving backwards, which we don't want. That would be reflux. But yeah, they do pass through the diaphragm.
Yeah, so we have like a hiatal hernia, that's probably stomach going through that.
Right, so a hernia just means soft tissue poking through a muscular wall. And so an inguinal hernia is when the intestines are poking through the abdominal or the pelvic wall at the inguinal area, the groin. And a hiatal hernia is when some of the stomach's poking through the diaphragm into the thoracic cavity there. So, yes.
Did we fix the hiatal one? Or did it just let it go?
I don't believe so. And mostly I say that because I have one. It was diagnosed when I was 26 and they never bothered doing anything about it. I just take omeprazole, and it limits acid production in my stomach so I don't get reflux. Considering that pharmacological intervention is rather successful, the risk of going in and doing surgery, especially at an area right next to the heart, is not a good idea. So yeah, I don't. Personally, I know I've not had it corrected, and I have cardiac issues, so they're not going to think about starting to, muck around in that part of my body anyways. But I don't think in general hiatal hernias are surgically repaired. Maybe they were at one time, but now there's drugs that can help with that. And before Nexium, Prilosec, that kind of stuff came along, people just take lots of Tums. So, other questions? Okay. So those two things kind of wrap up the anatomy of the system. And we'll take a break, and I want to come back and talk about the function of the system a little bit. And in doing that, kind of move through the different areas, sort of following what's going on in the book, and sort of highlight the functions that I want you to be aware of. Now, really, I would need more than half of one of our classes here, so more than an hour and a half to go through all of that probably. So I will be a little bit brief on some of those things, just pointing out what I want you to pay attention to. And you'll get there. Also, I usually do a rather extensive explanation of enzymes at the beginning too. I'm not going to do that here. I'll save it for the end, which corresponds a little bit more to how the book presents this stuff. And also so that if I'm running out of time, I won't deal with that so much. But I did make a recording of doing that for my 11 o'clock lecture. So you can go in and hear the full enzyme story there. I recorded it back on Tuesday. And speaking of recordings, I've done something new. I'm now posting my recordings as public. So as soon as I hit the stop broadcast button right here, like this,